So this is, again, a representation of a lipid bilayer. And what this is doing is describing the selective permeability of a lipid bilayer. So a lipid bilayer tends to allow things to easily cross it that have two properties. One of them is that they're hydrophobic. So they too are water hating, and therefore they tend to dissolve readily in the oily portion of the lipid bilayer. And the second thing is that they're not too big. So small hydrophobic things, water-hating things, examples being the oxygen that we breathe and the carbon dioxide that we breathe out, and also many drugs, because many drugs have targets that are inside of the cell. And in order for that drug to act on their target inside the cell, they have to get into the cell. And in order to get into the cell, without any facilitation of protein, because there's not necessarily going to be a protein around to take a specific drug into the cell, the drug on its own has got to be able to cross the lipid bilayer. So many drugs, in fact, are small and hydrophobic. So they have a high propensity to cross lipid bilayers. Also, small things that happen to be hydrophilic, that water-loving, are able to cross lipid bilayers. Not as effectively as the small hydrophobic things. An example here is this molecule urea, but more importantly is the molecule water. And in fact, in the lab, I'm not sure if it's going to be next week or the week after, we'll be looking at osmosis, which is the movement of water across membranes. Water very readily moves across membranes, not because water dissolves readily in the membrane. I mean, it does to a small degree, but because there's simply so much water on the outside of these membranes, because the membranes are found in a water solution. Over here, we have substances that are large or charged. Examples are the various ions, something like ATP, which we'll be talking about next chapter, and glucose. These things do not readily cross lipid bilayers. And then very large substances like proteins or starches don't cross lipid bilayers either. So we've got substances that very readily cross membranes, other substances that do so to a lesser extent, and then we've got everything else, and that everything else is very important. And the only way you're going to get these things across the lipid bilayer is if you have protein facilitation.